Hello everyone, I'm your YouTuber technician and I'm going to share one of the most popular instrument details with you guys. So stay tuned with me. So this is a PCR machine. A PCR machine, also known as a thermal cycler, is a lab instrument used for DNA amplification. The machine automates the temperature changes needed for the PCR polymerase chain reaction to amplify DNA segment. PCR test can detect disease when there is a only very small amount of pathogens in your body. Efficient use of PCR by the interprofessional healthcare team can lead to a early detection of bacterial and viral pathogens. So this is a PCR machine, also known as thermal cycler. PCR, the full form of PCR is polymerase chain reaction that amplify specific DNA sequence, creating millions of copies from same sample. This was invented by American biochemist Carey Mullis in 1983. So the procedure used to amplify it is amplification, molecular biology technique, DNA amplification, primers, cycles, and then applications. So it will not open if it is locked. This is the way it is locked. It will not open at this. It will not open at this. Only at 45 degree it can open. This provides high heat. This is a type of coil. Here we put the surface, turns very hot because it increases the temperature, and in these wells there are total 16 wells. Here we put the small tubes in that. This is a master cycler, and from these buttons we can set it. And now I am going to tell you the working of PCR machine. So first, setting up of the reaction. A small amount of DNA sample containing the target sequence is combined with primer DNA polymerase. This mixture is placed in a tube within a PCR thermal block. Here it is placed. This mixture is placed in tubes. So the first procedure this machine does is denaturation. The temperature raised to 95 degrees Celsius, causing the double-stranded DNA to separate into single strand. The second step is annealing. Temperature lowered up to 50 to 65 degrees Celsius to allow the primers to bind to their complementary sequence on the single stranded DNA. The third and the last step is extension. Temperature raised again at 72 degrees Celsius to allow DNA polymerase to extend primers, creating new DNA strands from that same DNA. These three steps are repeated multiple times, about 25 to 40 cycles. With each cycle, the amount of target DNA doubles, leading to exponential amplification. Final extension and analysis. After the final cycle, a final extension step may be performed, and the amplified DNA can be then analyzed using various techniques such as gel electrophoresis to visualize and identify the specific DNA sequence. The PCR machine provides the precise temperature control needed to drive the DNA replication process, enabling researchers to amplify specific DNA sequence for various applications including disease diagnosis, genetic research, and forensic analysis. This is the machine where the coronavirus amplification was performed.